wonder how DNA was actually discovered? Here's the truth. Nobody discovered it. They guessed. Scientists knew something like DNA existed, they just had no idea what it looked like. So they built wire models, hoping something would fit. It all started like this.
By the 1950s, scientists had figured out that a chemical called DNA carried our genetic traits. But when they tried to look closer, they hit a wall. To them, DNA was just a glowing, blurry mass. They were entirely blind to its physical structure. Imagine trying to understand how a complex Lego set is built without knowing the shapes of the individual blocks, let alone having the instruction manual. Without knowing the exact physical shape of the DNA molecule, understanding how it built biological life was impossible. The prevailing scientific method of the era was highly deductive. Instead of directly observing reality, the world's top chemists tried to guess the structure of DNA. They built massive, theoretical models out of metal and wire, hoping to stumble into a shape that made chemical sense. This speculative approach led brilliant minds to wildly incorrect conclusions. Linus Pauling, one of the greatest chemists alive, confidently proposed a triple helix structure for DNA. But chemically, the negative charges in the core would repel each other. A triple helix is physically impossible. The global scientific community was stuck. Theoretical model building had reached its limits. They desperately needed hard empirical data to anchor their theories to reality. Enter Rosalind Franklin. As a brilliant, uncompromising physical chemist, Franklin operated on strict inductive observation. Her philosophy was simple. Data must absolutely precede theoretical models. You don't guess the structure, you measure it. Her rigorous methods were forged during World War II. Franklin studied the microscopic porosity of coal. By understanding exactly how gases moved through tiny constrictions in carbon, she helped improve the design of gas masks to keep soldiers safe. Analyzing coal is difficult work. It forced Franklin to master a technique called X-ray crystallography, bouncing X-rays off molecules to map their atomic layout. She developed a rare, highly technical ability to capture clear images of microscopic, invisible structures. Franklin brought the tools of physical chemistry to a biological mystery. She treated the DNA molecule with the same systematic rigor she had used to map the pores of coal. In January 1951, Franklin arrived at King's College London on a fellowship to take over the laboratory's DNA X-ray diffraction work. She immediately ran into a severe culture clash with Maurice Wilkins, a scientist already studying DNA at King's. Wilkins possessed a totally different, more speculative scientific method, and mistakenly assumed this highly accomplished chemist was hired to be his assistant. While Franklin set up a quiet, meticulous basement operation in London, researchers James Watson and Francis Crick were up at Cambridge, engaging in loud, rapid-fire theoretical model building. Franklin ignored the guessing games. She refined a custom camera chamber, but allowed her to precisely control the humidity around individual DNA fibers, using different saturated salt solutions. That camera yielded an immediate breakthrough. Franklin discovered that DNA doesn't just have one shape, it exists in two distinct states depending on the humidity: a long, thin, wet form she called bDNA, and a short, fat, dry crystalline form called aDNA. Her rivals were playing with speculative metal puzzle pieces, hoping they fit. Franklin was systematically isolating the true physical properties of the molecule. From her crisp new X-rays, Franklin deduced that the strongly hydrophilic phosphate groups had to be on the outside of the DNA backbone. This single observation completely destroyed the previous theoretical models, which had assumed the phosphates formed a central core. Working with her assistant Raymond Gosling, Franklin focused her camera on the wet B-form DNA for a single 100-hour X-ray exposure. They labeled the resulting image Photo 51. But true to her philosophy, Franklin refused to publish her structural conclusions yet. She insisted on exhaustively reconciling the complex math between both the A and B forms before making any public claims. While Franklin was doing the math, Maurice Wilkins took her crystal-clear Photo 51 and showed it to James Watson. He did this without Franklin's knowledge and without her permission. Watch how this works. The specific X pattern in Photo 51 provided undeniable proof DNA was a helix, its angle revealing the exact pitch. A leaked report of Franklin's calculations established a 34 angstrom repeat distance and symmetry, confirming the strands run anti-parallel. Watson and Crick reached the finish line by assembling their model atop the exact empirical blueprints Franklin had painstakingly engineered. In April 1953, the journal Nature published 3 articles. Watson and Crick claimed the double helix structure in the lead paper. Franklin's supporting data was relegated to the back, making her foundational, original work look like a mere confirmation of their theoretical genius. Franklin was largely unbothered by the race for fame. She was just relieved to leave the toxic environment of King's College and transfer her research to Birkbeck College. At Birkbeck, she pivoted to studying the tobacco mosaic virus. She took her exact same rigorous X-ray methods and applied them to a biological structure far more complex than DNA. Here, she achieved her final major breakthrough. Franklin proved that the RNA in the virus wasn't sitting in a hollow cavity. It was a single-strand helix embedded directly within the viral protein structure itself. Tragically, Rosalind Franklin died of ovarian cancer in 1958 at just 37 years old. 4 years later, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize. Franklin's legacy is defined by her methodology. By prioritizing hard measurement over theoretical guessing, she established the operational standard for all modern structural biology.
If this story mattered to you, don't let it disappear into the archive. Hit like, subscribe, and help bring the next forgotten genius back into the light. Still have questions about how this all really happened? Leave them in the comments. The real answers are far more interesting than the official story.